And yeah, again, feel free to jump in with a question. You're going to have to say just interject verbally, and sometimes I tend to speak pretty fast. So I will not be insulted if somebody jumps into the middle of a sentence that I'm saying. So today I'm going to talk about the assembly, structure, and mechanobiology, a little bit about the mechanobiology of plant cell walls. So I've been at Penn State for a little over a decade now, and I'm excited to talk to you all today. So one thing I want to emphasize before I get started is that the plant cell wall is a very dynamic structure. We often think of plants, or I thought of plants when I was an animal cell biologist before I got into plants, as sort of static and not doing a whole lot. On the left is a little video of the process of cellulose being made, and on the right is a pair of guard cells that are opening and closing. So I'll touch on each of those sets of dynamic processes throughout the lecture today. And I think I might actually just switch my laser pointer back to the regular cursor because my laser pointer is not allowing me to advance the slides. Gotta love Zoom. Okay, so can everybody still see my arrow cursor if I move it around?
Yes.
Great, okay. So why, as somebody who started out as an animal cell biologist, did I choose to study plant cell walls? One of the motivations is that we rely a lot on fossil energy sources still, although renewable energy is growing as a portion of our energy use here in the US and around the world. But those fossil energy sources contribute to climate instability, and they're going to run out. The graphic on the lower left here is showing how much energy there is total in reserves of things like natural gas, petroleum, uranium, and coal, versus how much solar energy hits the planet every year, right? It's 23,000 terawatt years per year. And this is how much the world uses every year, at least as of 2009. So solar energy is really abundant, and plants are able to capture some of that solar energy and store it in the form of polysaccharides and other components of their cell walls. So plants generate about 185 gigatons of biomass every year. This is just land-based plants. There are also aquatic plants and related algae. And of that, a pretty large proportion, about 145 gigatons, is composed of cell walls. We use about 2 gigatons of cell walls every year for all purposes, things like building materials, fuels, things like that. So, you know, obviously the rest of this is part of the natural ecology of the world, and we don't want to completely disrupt that or co-opt it for human use. But one of the rationales is that we could potentially increase the proportion of cell walls that we use on an annual basis without, you know, completely destroying the ecological system. So one of the other great things about using plant cell walls to make bioenergy, as opposed to things like corn, is that we can actually generate net negative greenhouse gas emissions. So this is a graph where we're comparing the total greenhouse gas emissions as a percentage of petroleum. So petroleum is at 100%. Corn ethanol, which is the primary biofuel that's used in the US today, generates about 80% of the greenhouse gas emissions of gasoline. So it's better, but not that much better. Sugarcane is substantially better. It generates about 40% of the emissions. But if we were to convert something like switchgrass, which is a native grass in North America, into ethanol, we could actually get net negative greenhouse gas emissions. So because I want today's lecture to be a little bit interactive, I'm going to ask people to hypothesize why that might be the case. Why would we get negative greenhouse gas emissions, accounting for all of the growth, harvesting, transport, conversion, and then ultimately burning that fossil or that biofuel? You know, why would we end up with negative greenhouse gas emissions? So any thoughts?
Is it because you're using plants? So technically you're putting more plants into the world before you take them out?
Right. So we grow the plants and then we harvest them, and ultimately we sort of burn them indirectly in the form of biofuels. So we are growing plants, but we harvest a lot of that plant biomass. So, you know, you could think about that being maybe a net zero, but why would we have net negative greenhouse gas emissions from switchgrass?
Could the product be used as nutrients for other plants?
Okay, so we could potentially recycle some of that product back into something that could be used by other plants. And that's kind of correct in the sense that plants actually do that for us to some degree. So when we harvest things like switchgrass, we're harvesting the above ground part of the plant. But switchgrass, if you look at it in total, looks like this. So it has a pretty large root system, a couple of meters long root system, and then about a meter and a half of above ground growth. So this top part is the part that we would harvest to convert into biofuels or other products. And then the plant is generating, capturing carbon, generating polysaccharides, and then moving some of those polysaccharides into its root system to generate plant cell walls down here and tissue down here. And this root system helps enrich the soil. It helps support the microbiome in the soil. And some of that carbon is cycled back into the atmosphere over time, but that actually happens on a very slow timescale, on century timescales. So when we think about things like carbon capture and sequestration or direct air capture, plants are already doing that for us, right? They're capturing CO2 from the atmosphere, converting some of that CO2 into sugar, pushing that sugar underground and burying it for us, and doing this on gigaton scales. So this also enriches the soil and helps improve agricultural yields. So plants are proposed to be one of the potential solutions that will help address this challenge of climate change if we can do bioenergy and biofuels responsibly and correctly. Okay, so cell walls serve a whole lot of different functions in plants. They help determine the size and shape of plant cells, right? Most of which are surrounded by a plant cell wall. They support upright plant growth. So this is a picture I took out in Muir Woods in California. It's right north of San Francisco. These redwood trees are about 100 meters tall, and they're composed mostly of sugar, which is pretty amazing to me. That sugar takes the form of polysaccharides that help support the upright growth of these plants. Plant cell walls also help the plant prevent water loss from its surface, especially some of the more waxy and hydrophobic parts of the plant cell wall. They protect plants against pathogens and environmental stresses. So plants do have an innate immune system. They don't have an adaptive immune system like animals do, but they have an innate immune system, but the cell wall forms a barrier against pathogens and other stresses. And then importantly, for addressing the climate challenge, they contain much of the carbon that's fixed by photosynthesis. So we can potentially use plant cell walls as sources of food, materials, and bioenergy. So this is just a little cartoon structure of a plant cell. One of the other interesting things about plant cells is that they tend to have quite large vacuoles. So they have a cytoplasm, a nucleus, and then a vacuole that can take up to 90% of the volume of that interior part of the plant cell. And then that's all contained in a plasma membrane. And that plasma membrane is surrounded by two layers, in some cases, of cell wall. One is called the primary cell wall that's laid down while the plant cell is growing. And the second layer is called the secondary cell wall that's laid down often as the growth of that cell is either decelerating or has stopped completely. And this compartment outside the plant cell, we plant biologists call the apoplast, right? As opposed to the cytoplasm. So the apoplast is this kind of unique metabolic compartment that contains a bunch of polysaccharides, some structural proteins, some enzymes, some small molecules that we don't really fully understand what they're doing out there. But I like to think of it as sort of like the desert of the plant tissue, right? It doesn't contain a whole lot of ATP because ATP is produced intracellularly. It doesn't have a whole lot of metabolic activity, but it does have some limited forms of metabolism. So when we think about these primary and secondary cell walls, we can categorize the components of them. Some are common to both primary and secondary cell walls. So I'm going to introduce these in more detail in just a minute. But we can think of cellulose as being one of the most important parts of the plant cell wall. Hemicelluloses, glycoproteins, and metabolites. Primary cell walls are often enriched in another form of polysaccharide called pectins. We have enzymes and a whole lot of water in primary cell walls, whereas secondary cell walls also contain two compounds called lignin and silica. And this lignin and silica is thought to strengthen the secondary cell wall and support upright growth in plants. So when we think about the biosynthesis of cell wall components, plant cells have a Golgi apparatus, or in most cases, several Golgi apparatus that move around in the cytoplasm. So they're a little bit different from the Golgi that exist in mammalian cells. where that's clustered around the centrosome, which is what I did my PhD on back in the day. But these Golgi, I like to think of as sort of mobile factories for the components, some of the components of the plant cell wall. So the Golgi contains these cis-medial and trans cisternae, and they help deliver proteins called cellulose synthase complexes to the plasma membrane. And cellulose is actually extruded directly into the cell wall at the cell surface by this cellulose synthase complex. Whereas some of the other components of the cell wall, hemicelluloses and pectins, for instance, are made in the Golgi and then trafficked out to the cell wall. So for instance, we can have a hemicellulose synthase that might be resident in the membrane of one of the Golgi cisternae. It helps to polymerize cellulose, or that hemicellulose, which is then trafficked through the trans-Golgi network into post-Golgi trafficking vesicles, which fuse with the plasma membrane and deliver that hemicellulose out into the apoplast. So this process of cell wall assembly involves not only the synthesis of certain polysaccharides directly into the wall, but also delivery of pre-synthesized components out to the wall, and those come together to form the entire sort of meshwork of the plant cell wall. We also have proteins that are secreted through a canonical secretion pathway that starts actually when they're synthesized on the surface of the rough ER, delivered through the Golgi, and then trafficked out to the apoplast. And then likewise with lignin, for instance, in secondary cell walls, we have monolignals. These are the monomers that make up lignin ultimately, and they're made metabolically in the plant cell wall from amino acids, and then are thought to diffuse through the plasma membrane out into the wall where they polymerize. Okay, so one of the most exciting processes, and really this is kind of one of the, sort of most abundant metabolic processes on the face of the planet, because cellulose is the most abundant biomolecule in nature, is the process of cellulose synthesis. So plant cells, unlike animal cells, which have their, most of which have their microtubules organized around the centrosome, most plant cells lack a centrosome, and they have their microtubules arranged in arrays along the cell cortex, so along the edge of the cell. And these microtubules help guide these cellulose synthase complexes that move, as you can see in these little green dots that are moving along the surface of this cell. They help, the microtubules help guide these cellulose synthase complexes as they're spinning cellulose out into the wall. And we think this process is helping to drive the pattern deposition of cellulose in a certain direction that gives the wall what we call mechanical anisotropy. So if the cellulose were arranged randomly in the wall, we could think of that cell wall as being potentially isotropic in nature, meaning that it can expand in every direction equally. But in many cases, the cellulose is sort of wrapped around the cell wall. And the analogy I like to use is a slinky. So a slinky can expand in this direction very easily, but if we turn it on its side, it can't expand in this direction very easily because of the nature of that spring being wrapped around radially. So the same thing happens with cellulose in many types of plant cells and constrains the direction of growth of that cell and also the mechanical properties of the wall. So cell wall composition not only varies between primary and secondary cell walls, but it varies across different plant species and even tissues within those plant species. So one of the main divisions of flowering plants is eudicots or dicots versus monocots, which include grasses. So in dicots, there tends to be more pectin in the primary cell wall. In monocots, there tends to be less pectin and a bit more hemicellulose. There's roughly the same amount of cellulose in both. And there's some number of cell wall proteins, which are more abundant in dicot primary walls. And then in secondary cell walls, again, we have this lignin compound, this polyphenolic lignin, and there's some amount of silica in both dicot and monocot cell walls. You can see that the cellulose is more abundant in dicot secondary cell walls. Okay, so what are the building blocks of plant cell walls? We can think of monosaccharides, many of which are generated through central metabolism, as being these basic building blocks. So we have hexoses, which contain 6 carbons each, glucose, galactose, and mannose, for instance. We can have pentoses like xylose, arabinose, and apiose. Uronic acids, which are acidic monosaccharides that have this carboxyl group that can either be protonated or deprotonated depending on the pH of the medium. Those include galacturonic acid and glucuronic acid, and then deoxysugars like rhamnose and fucose, right? So these are hexose sugars that are that from which an OH group is removed in one of the carbons. So these are the basic building blocks that plant cells use to generate the polysaccharides in the cell wall. But in many cases, they have to convert these sugars into an NDP, or nucleotide diphosphate form of the sugar, in order to allow it to be acted upon by a glycosyl transferase to polymerize into the polysaccharide. So the monosaccharides by themselves are not especially metabolically labile, whereas these NDP forms of the sugars are what really are the substrates for making the polymer. Okay, so what is cellulose? Cellulose is a cable of glucose chains, which we call glucan chains, that have this beta-1,4 linkage. So if you remember back to OChem, some of you might have, nightmares about OChem, but it's okay. We won't be testing you on OChem today. This beta-1,4 linkage is between the 1 carbon and the 4 carbon of sequential glucoses in the glucan chain of cellulose. And this planar configuration of the beta linkage means that the glucose rings, right, this is sort of a chair configuration, but the glucose rings in this case are roughly parallel to one another, but you can see that they're flipped, right, 180 degrees with respect to this C6. So the C6 is on the top here and on the bottom here. And this means that cellulose is actually made of alternating monomers of glucose. And together, these two glucoses together form what's called a cellobiose unit. So this is a 2-glucose unit that's the repeating unit of the cellulose itself. So these glucans form long, multiple hundreds or even thousands of glucose chains. And those chains are then hydrogen bonded together in a way that resembles a cable into different forms of cellulose. And there are a bunch of different so-called allomorphs of cellulose. I'm not going to get into the details of that, but we can think about those glucan chains, which we're now looking at in sort of an end-on cross-section, as potentially containing different configurations, right? So they can have, for instance, a 2-glucan, 3-glucan, 4-glucan, 4-glucan, 3-glucan, 2-glucan. So a 2-3-4-4-3-2 configuration, a 3-4-4-4-3, or a 3-3-3-3-3-3-3. So these are different hypothetical configurations of the glucan chains in the cellulose, and there's actually debate still in the scientific community about what that configuration is in real life. For a long time, cellulose was thought to have 36 chains and be quite large in its cross-section, but more recently, there's been a hypothesis that it contains somewhere between 18 and 24 chains in cross-section. So cellulose has this amazing property that it has very high tensile strength because of the strength of this glycosidic bond and also the hydrogen bonding that exists between glucan chains in the cellulose crystalline configuration. So its tensile strength is similar to that of steel, and that's part of the reason that we make a lot of our clothing out of cotton, which is primarily cellulose. Okay, so around that hemicellulose and interlinking the cellulose in the wall is a set of polymers called hemicelluloses. Sometimes these can actually have the same beta-1,4 glucan backbone that cellulose does, but they're often modified with side branches. So this is xyloglucan, which is the most abundant hemicellulose in dicot primary cell walls. It contains side chains that can have xylose, galactose, and fucose. We can have mannans that can be galactosylated to form galactomannans, and they can also be interspersed with glucose to form glucomannans. There's a form of hemicellulose called beta-1,4-1,3 mixed linkage glucan. So that contains beta-1,4 linkages that are interspersed with beta-1,3, which provides a kink in the backbone of the mixed linkage glucan. And then we have xylans, which are the most abundant hemicellulose in the primary cell walls of grasses. And these xylans have a beta-1,4 linked xylose backbone. Remember that xylose is also it's a pentose sugar rather than a hexose sugar, but it resembles glucose in many ways. And that can be modified with glucuronic acid and arabinose side chains. So together, these hemicelluloses, we think, provide sort of an intersperser that prevents the cellulose in the wall from all bundling together and forming sort of a giant bundle. And then the so-called matrix of the cell wall is composed primarily of pectins. And because these pectins contain galacturonic acid and in some cases glucuronic acid, these are acidic sugars that I mentioned earlier, these can be charged and they can also be methyl esterified. And pectins tend to be highly hydrated. So you might have encountered pectin if you had jam or jelly with breakfast this morning or recently. That is primarily pectin. So it's a very hydrated gel, and that provides some flexibility and fluidity to the cell wall, we think. There are three different major domains of pectins. The most abundant in some cases is called homogalactronan. It's a linear polymer of galactronic acids that are linked by alpha-1,4 linkages this time. And its carboxyl groups can be methyloesterified or demethyloesterified. If they're demethyloesterified, the pectin becomes negatively charged and can be cross-linked by calcium. And then there are two other forms of pectins, rhamnogalactronan 1 and rhamnogalactronan 2. RG1 has an alternating backbone of galactronic acid and rhamnose residues, hence the name rhamnogalactronan, and it contains arabinan and galactan containing side chains. And then rhamnogalactronan 2 is this pretty complex molecule that has a variety of different side chains, up to 6 different types of side chains. And one of these side chains, chain A, contains apiose residues that allow for cross-linking via what are called borate diesters. So one of the key sort of features of pectins is that they are, in many cases, reversibly cross-linkable in the wall, and they provide strength to the wall in a way that's different from the hydrogen bonding of cellulose or the interactions of hemicelluloses. So pectins can be covalently linked to one another in certain domain configurations. You can have homogalactronan, RG1, and then some more homogalactronan, or likewise with RG2. And it can also be connected to arabinogalactan proteins, which are a form of glycoprotein. So there's one hypothesis in the literature that pectins are actually made as sort of complex glycosylation subunits of glycoproteins, and then those pectins are cleaved off in the wall. But that's a bit controversial, and we don't really know exactly how these pectins are polymerized in the Golgi. Okay, so the final major polymer that I'll talk about as a wall component is lignin. It's a polyphenolic compound that's composed of 3 major monolignal subunits. These are called p-coumaryl alcohol, coniferyl alcohol, and synapyl alcohol. And these come together to form different subclasses of lignin called S, G, and H lignin. So you can see that this is a phenolic ring, right? The OH and the benzyl ring form a phenol group. And they also have this modification that's opposite the OH group in the phenolic ring. So these monolignals can be acted upon by enzymes in the wall that form them into what are called monolignal radicals, right? They cause them to have single electrons. And those single electrons then allow these monolignals to react with one another to form beta ether and ester linkages that generate this fairly complex polymer that has these, again, these phenol groups interspersed. And this you can imagine, again, thinking back to OCHEM, that this whole polymer might be fairly hydrophobic just due to having all of these phenolic rings as part of it. And that's the case. And we think that lignin provides sort of a waterproofing function to the wall and also enhances the strength of the plant cell wall. So this is laid down in secondary cell walls, and it's part of the reason that wood, like the bookcase behind me, right, is brown. Lignin itself is brown, and also part of the strength of woody material. Okay, so cell wall proteins can include structural proteins like glycoproteins that I mentioned, and also enzymes. So glycoproteins have complex sugar moieties that are linked to hydroxyproline residues that are part of the amino acid sequence of the protein. And then we can also have non-structural proteins, things like expansins, which my colleague in the CMB here at Penn State, Daniel Cosgrove, has studied for quite some time. We can have transferase enzymes that actually cleave and relink sugars or polymers back together, hydrolases, lyases, esterases, and oxidoreductases. So the plant cell wall is quite active enzymatically, but you can see that most of these are not glycosyltransferases. They're not acting on a small substrate and doing polymerization in the wall, but rather, in most cases, either cleaving and then relinking wall polymers. One thing I wanted to emphasize is that water is actually an important part of the cell wall, especially in primary cell walls. So we think that, for instance, that water provides a boundary layer between xylan, this hemicellulose in the wall, and cellulose. It's probably involved in some of the hydrogen bonding that gives rise to adhesion of that xylan to the cellulose layer. And in this again, cross-section of a bundle of cellulose that's surrounded by mannans in this case, you can see that the water, which is in green here, is sort of surrounding and interspersing and interlinking all of these wall polymers, including lignin, hemicellulose, and cellulose. So when we, for instance, dehydrate and then rehydrate plant cell walls by solid state NMR, we can detect changes in the nanoscale architecture of the cell wall that imply that hydration is actually a really important part of how that cell wall is assembled. So even though we might think of something like wood as being dry, it actually contains 10 to 20% water in most cases, unless we like really kiln dry it. And when we do that, we're actually changing the structure of the plant cell wall quite dramatically. Okay, and then there are some limited small metabolites that are present in the wall, monosaccharides that are the products of wall breakdown. We can have ions, things like calcium in particular are quite important for cross-linking pectins. We have redox compounds, hydrogen peroxide, superoxide, radicals, ascorbic acid, and then also acetate ferulic acid and many, many other small molecules that are, again, mostly the products of breakdown of those cell wall components rather than being synthesized directly in the wall. Okay, so what does the primary cell wall look like? This is a diagram that's now a full 20 years old, but still pretty relevant to our understanding of how the primary cell wall is composed. We think of the cellulose in the wall as being sort of like these, again, these cables that are composed of glucan chains that are put together and polymerized by the cellulose synthase complex. And these long cellulose so-called microfibrils are interspersed with hemicelluloses. So those hemicelluloses lie along the cellulose, cables, and then they also might actually interlink different cellulose microfibrils to each other. And then this is all embedded in a pectinaceous matrix. That pectin is shown in sort of blue and purple in this diagram. And I should also note that in this sort of artistic diagram of the primary wall, all of the polymers are 30 times less dense than they are in real life. So I like to think of the plant cell wall as a giant bowl of a bunch of different types of pasta, like linguine and fettuccine and spaghetti and maybe angel hair pasta, all mixed together, right? And all interacting in very interesting ways. So another thing I should point out is that there's a special layer of the plant cell wall called the middle lamella that's highlighted in yellow at the top of this diagram. And that's a pectin-rich layer that helps plant cells adhere to one another. We think that this is really important for maintaining the positions of plant cells in the plant body, right? So unlike animals, which have cells, certain cells that migrate with respect to one another, most plant cells are locked in place after cell division. And it's partly because of that adhesive middle lamella that keeps them in place. I added this diagram of the secondary cell wall, and we can think of this is a sort of molecular scale cartoon, I'll say. It's not, based completely on data, but it's inspired by our understanding of the interactions between these polysaccharides. In the secondary cell wall, we can think of the cellulose microfibrils, which again are these sort of 2, 3, 4, 4, 3, 2 configurations as bundling together into larger, what are called macrofibrils. And those can be surrounded by hemicelluloses that are shown in yellow here. This is an example of xylan, for instance. And then lignin sits on the surface of all of this. That's the stuff that's shown in brown in this diagram. So secondary cell walls, again, contain much less water because of the presence of lignin. They contain more bundled cellulose, much less pectin than primary cell walls, and they're probably more static and also just structurally stronger than primary cell walls. So thinking a little bit about the mechanics of plant cell walls, one of the really interesting things about them is that they can exhibit both elastic, plastic, and viscoelastic mechanical behaviors. So I'll just pause for a second and ask if anybody knows the definition of elastic and plastic. If you've done any mechanics work in your previous lessons. So what's an elastic behavior?
Deformation that's recoverable.
Yeah, so it's deformation that if you stretch it and then you release that stress, right? You're applying, let's say, tensile stress on an object. you release that stress, it returns back to its original state. So I'll just pretend that this, I mean, the slinky is sort of somewhat elastic, but if I stretched it out, right, and then I released the stress, it tends to go back to its original state. Okay, so what's a plastic behavior?
Permanent deformation.
Yeah, so when I deform that material, it stays deformed, right? So you can think about that like the difference between a rubber band, which exhibits a lot of elastic behavior, and something like a piece of taffy. So if we pull that taffy, it tends to stay stretched out, right? So that's a plastic behavior. And then viscoelastic mechanical behavior is elastic behavior that contains either a time lag or resistance to that elastic deformation that changes the time component, the time dynamics of that elastic response. So plant cell walls, we think, have this kind of viscoelasticity. They also exhibit plastic and elastic behavior. So they have complex mechanical responses to tensile stress, for instance. So this is a mechanical test of an onion epidermal peel, which is a really classic system for studying cell wall mechanics that, again, was developed by the Cosgrove lab here at Penn State. So you peel the epidermal surface off of an onion scale, and they clamped it in between these two holders. And they're watching what happens when they apply tensile stress to this material. So you can see that it's stretching out, and it's exhibiting some pretty interesting behaviors. First of all, it starts to flatten a little bit as it's stretched out. And then it starts to wrinkle a little bit and become more opaque. And they're doing this in a cyclic way. So they're cyclically loading and unloading, right? They're stretching and unstretching the onion material. And from measuring both the relative length and the actual mechanical stress that's being placed on the tissue, they can derive interesting information about how that plant cell wall material, in this case, it's actually just a single layer of cells that are connected to each other, but have properties that are determined by their walls. They can measure interesting changes in the mechanical response of that material over time or over different cycles of mechanical loading. So Jingyi Yu, who's one of the CEMB postdocs working in Dan Cosgrove's lab, has done some really exciting mechanical testing of the onion epidermis and has a really nice PNAS paper that came out just this spring that talks more about that. Okay, so viscoelastic deformation is a little bit harder, at least for me, to understand intuitively. So one of the sort of nice ways that I like to think about it is it's sort of like the combination between a spring and a dash pot. So a spring, we've all encountered a spring at some point in our lives. A dash pot you can think of as sort of like a syringe, right? So if you're using a syringe, let's say to inject something, a dash pot has resistance to, it has a viscous resistance to tensile deformation. And you can think of the cell wall as being a combination of a spring and a dash pot. That's where the viscoelastic behavior derives from. So ultimately, the deformation of the plant cell wall is driven by turgor pressure. So plant cells have the ability to, for instance, pump hydrogen ions across the plasma membrane using proton ATPases. So they consume ATP to pump protons across the plasma membrane. And that can allow the membrane to hyperpolarize. And that hyperpolarization will then open voltage-gated channels that allow ions into the cell. So things like potassium ions in particular. So when those potassium ions enter the cell, it creates an osmotic potential across that plasma membrane that causes water to ultimately enter the cell. So plants are generating this turgor pressure by consuming or burning ATP, and that turgor pressure then pushes outward on the cell wall that's encasing that plant cell. So that's we think one of the driving forces that ultimately causes the deformation and expansion of the wall. So as that turgor pressure increases, the wall ultimately will undergo what we call stress relaxation, meaning it will change its configuration, right, to allow for that turgor pressure to go down and to relax the stress that exists in the wall. One of the other important processes that has to happen during this to allow the cell to actually increase in volume is enough water has to enter the cell to take up that new volume, right? So water uptake and expansion is thought to be part of this cycle of pressurization, stress relaxation, more water uptake, maybe due to additional ATPase activity, And that cycle continues to allow the plant cell to ultimately grow and expand. And plant cells are really good at expanding. In many cases, they can expand over 100-fold in volume as they're growing. So I mentioned earlier that the cell wall is often anisotropically configured, especially the cellulose being wrapped in a particular direction. This gives the cell wall this anisotropic mechanical response. So if we think about turgor pressure pushing outward equally in all directions, the constraint that's imposed by the cellulose that might be wrapped around the cell in this direction, for instance, will then cause the cell to elongate in this direction while not expanding too much in this direction because of that cellulose being wrapped around it. So this anisotropic organization of the wall is one of the things that gives different plant cells different shapes. And plant cells can have all sorts of different shapes. They can be cylinders, they can be puzzle piece shaped, they can form these like cool spiky branch configurations, these trichomes on the plant surface. And that's all driven, we think, by how the cell wall is laid down to respond to the isotropic turgor pressure that exists inside the cell. So one of the sort of big advances that's happened over the past few years in our mechanical understanding of plant cell walls is that the Cosgrove lab, in collaboration with Su-Lin Zhang's lab here at Penn State, developed what they called a coarse-grained model of the onion cell wall. So they had done a lot of mechanical testing and atomic force microscopy, scanning electron microscopy to understand the molecular architecture and the mechanical responses of onion cell walls. And they then took that information along with information derived from molecular dynamics simulations of different wall polysaccharides and generated an in silico, an in the computer version of the multilamellar onion cell wall. So when I say multilamellar, I mean that we have multiple layers of cellulose that are oriented in different directions. So I'll go back just for a minute to this primary cell wall diagram. You can see that in this case, we have a layer of cellulose that's arranged in this orientation, and the second layer is arranged 90 degrees relative to that, and then the third layer is 90 degrees. This isn't quite accurate. This is one of the few things about this model that's not accurate. The cellulose tends to be arranged in sort of roughly 60-degree arcs relative to the previous layer. So that's what they are modeling in these 4 different layers of the cell wall. And they generated this sort of, cell wall patch, and then they were able to computationally stretch that cell wall in certain directions. So when they did this, and when I say coarse-grained modeling, I mean they're not representing each of the molecules as its individual atoms, but rather as a series of connected beads, right? Sort of virtual beads. And the reason for that is that it's computationally too expensive to build a molecular architecture of the cell wall at the atomistic scale and then do things with it. It just becomes computationally prohibitive. So you have to sort of simplify the representation of the wall into this coarse-grained configuration to then look at the mechanics of a big enough piece for it to make sense and connect to what you see experimentally. So when they did this, they saw that, and for instance, they stretched the wall in this direction, they saw that the cellulose that was arranged closest to the stress direction underwent a lot, or experienced a high level of stress, which you might expect, right? It's potentially sort of resisting that deformation in this side-to-side direction the strongest. Whereas the cellulose that's arranged more perpendicularly to that stress direction is not experiencing a whole lot of stress. So one of the things that they observed when they did this sort of in silico stretching experiment is that the cellulose tended to slide, right? The cellulose, first of all, tended to bundle a little bit, even in the primary wall, and then that cellulose tended to slide relative to itself. And that sliding behavior, they think, is one of the things that drives the plastic deformation of the plant cell wall. Because you can think of, let's say just a polymer that's kinked and then it gets stretched out in this direction. If it were to rekink, that would be an elastic behavior. But if you have sliding of polymers relative to one another, you can think thermodynamically, it's not so favorable for those polymers to then slide back, right, relative to one another into the same configuration. So the thinking is that that cellulose sliding is one of the molecular mechanisms that's underlying this plastic deformation. So this study really highlighted the central role of cellulose in determining the mechanics of the plant cell wall, at least in the onion epidermis that they were modeling in this particular study. So Yao Zhang, who's the former postdoc from the Cosgrove lab, who was the first author on this paper, now has its own group in China and is doing kind of this coarse grain modeling for all different types of cell walls and trying to understand how they work. So it's pretty exciting stuff. Okay, so I wanted to pause there because I gave just a brief taste of sort of cell wall mechanics as we understand it at the, I would say maybe non-biological level, and see if anybody had questions so far. Because I want this to be a little bit interactive. So please jump in with questions if you have them.
I have a question.
Yeah.
It goes back to the response to like the cell wall whenever there's like an influx of water and stuff like that. You were mentioning that like it increases in size, but I thought like that at least the primary cell wall couldn't increase in size. Or does it also change in response to like the water influx?
Right, that's a really great question. So the cell wall, the primary cell wall does increase in surface area. It increases in size. And that can happen via a couple of different mechanisms. One is that the polymers can actually get stretched out. So we notice, for instance, when we look at like a growing so-called hypocotyl, right? This is sort of you can think of an alfalfa sprout as being basically one large hypocotyl. When those cells are elongating, there's not a whole lot of new wall being laid down. And instead that wall is just getting stretched out and getting thinner. And that stretching and sort of reconfiguration of the wall polymers, they might sort of go from being roughly this direction to being stretched out in this direction as the whole cell gets elongated. We think that involves just sort of geometric reconfiguration of some of the wall polymers and the overall thinning of that wall as it gets stretched out. So that's one of the mechanisms of cell wall enlargement or expansibility. And then the other thing that's happening in some tissues is you have new wall polymers like cellulose, hemicelluloses, and pectins getting delivered out to the apoplast as the cell is actively growing. So unlike something like a crab shell, right, which is laid down when the crab molts, right, a new shell is laid down and then the crab starts growing, and eventually the crab has to molt again to continue its growth, right? This is the case where you have layers of cell wall that are being added interiorly as those older layers are being stretched out and maybe thinned to some degree and maybe even sort of breaking apart. So I think of the plant cell wall as kind of like an exoskeleton that can grow along with the cell that it contains, which is pretty cool to think about. And then secondary cell walls we think are laid down mostly after cell growth has completed. So as that thicker layer of secondary wall gets laid down, in some cases it gets so thick that it actually sort of compresses the cell into a tiny little chamber in the middle of that cell. That's especially true in things that are called stone cells or sclerids. So if you've ever eaten a pear and felt that sort of little grittiness in the pear, those are actually little sclerids or stone cells that have very, very thickened secondary cell walls, almost to the point that the cell is like really gone. And often after secondary wall synthesis is complete, that cell will die. So especially in the case of vessel elements that conduct water through the stem of a plant or the trunk of a tree, that secondary wall is laid down and then the cell inside dies and it becomes a hollow tube through which water can be transported effectively. So we don't fully understand all the mechanisms by which the cell wall both grows and is synthesized at the same time. That's one of the things that my lab is interested in, kind of combining this mechanical approach with an understanding of the cell biology that's happening inside to generate new wall polymers and change the configuration of the wall. But yeah, it's a good question.
Thank you. That's really cool. Can I just do a follow-up really quickly?
Fire away.
I was like, thank you, but now I have a follow-up question.
Awesome.
So if the primary cell wall is the one that I forgot, has the pectin, P-E-T-I-N.
Pectin, yeah.
Pectin, yes, if that one's the one that has a pectin, if it does go through the process of like becoming thinner and stuff like that, is there like a trade-off between like cells being able to attach to each other as they grow? Like, do they have like weaker attachments as they grow?
That's a really good question. So how do cells stay stuck to each other? How do plant cells stay stuck to each other? The thinking is, well, my thinking is that some of the pectin that's in the primary cell wall as that wall is getting thinned out actually gets squeezed into the middle lamella. one analogy, this is maybe not a perfect analogy, but one analogy I like to think of is kind of like if you like squeezed a bunch of toothpaste into a sponge and then you squeeze that sponge, the toothpaste is slowly going to squeeze out of that sponge. And that toothpaste is what my analogy of pectin, right? That could help maybe stick two sponges together. Again, not the perfect analogy. And it sounds kind of gross with toothpaste in a sponge. But anyway, we think that pectin is providing that adhesive layer between the cells. So when we look at certain mutants that are defective in the production of pectin, for instance, their cells actually start to delaminate as the plant is growing. So they have cells that come apart. Shockingly, the plant is able to survive that, partly because it has other enzymes that make pectin later in its development. So it's not lacking cell adhesion throughout its entire development, but in seedlings, at least, it has cell adhesion defects. This is one of the things that differentiates plants from animals, right? Plants can have all sorts of damage happen to them and survive because they have many of their cells are capable of acting as stem cells and producing new tissues. That's why you can take like a leaf cutting and generate an entire plant out of that leaf. But yeah, there are instances in which the cells actually separate in a developmentally controlled way. So you can think of every autumn, here in the Northeast, the leaves of deciduous trees fall off, and that's actually a degradation of the pectin in that middle lamella layer at the base of each leaf to cause the leaf to fall off. So there are controlled instances in which the cells actually do separate, and that's beneficial to the plant. But the thinking is that cell adhesion in most cases keeps these cells locked together.
Thank you.
Sure thing. Any other questions? Okay. So I'm going to jump into a little bit of mechanobiology that deals with something that I don't study in the CEMB, but another part of my lab studies. And these are stomatogard cells. They're some of my favorite cell types in plants. Stomata are pores on the surface of plants that can regulate the uptake of CO2 and the transport of water from the roots up to the aerial tissues. So each stomatal pore is surrounded by two, in most cases, guard cells. And these guard cells in eudicots are kidney-shaped. And they can deform to allow for the opening and closing of the stomatal pore. So they expand and elongate to open the pore. They actually push against each other to open the pore. And then they can shrink and lose water to close the stomatal pore. So these stomata are responsive to environmental conditions, and they allow the plant to carry out photosynthesis by taking up CO2 without losing too much water, or they allow the plant, again, to transport water from its roots up to its shoots. So in these guard cells, these stomatal guard cells, the cellulose is wrapped radially around those cells, and we have studied this configuration of cellulose in both open and closed pairs of guard cells, which is what's shown in this little sort of fake movie here between the open and closed state. And if you look at this for a while, you'll see that in the open state, the cellulose is sort of evenly distributed across the wall, whereas in the closed state, it tends to be sort of arranged in these little fibers that you can make out. So we think that implies that the cellulose is actually changing its configuration between the closed and the open state of these guard cells. Okay, so one of the things that we study in these guard cells is pectin. And again, as a reminder, the simplest and most abundant pectin in most cases is homogalactronan. So to get into the details of how homogalactronan can be modified in the wall, it's synthesized in a methyl form. It has these CH3 groups on its carboxyl units. And these methyl ester groups can be removed in the cell wall by enzymes that are called pectin methylesterases. So these pectin methylesterases remove the methyl group and they expose these negatively charged carboxyl units on the galactronic acid residues. Those negatively charged carboxyl groups can cross-link with calcium to form a strong gel in the wall. And we think that this mechanically strengthens the wall. Or if the methyl ester groups are not completely removed, allowing for efficient cross-linking with multiple atoms of calcium, in some cases, the pectin becomes susceptible to cleavage by enzymes that are called polygalactronases or pectate lyases, depending on their enzymatic mechanism. So I like to think of these as little molecular scissors that cleave pectin in the wall and make it into shorter polymers. So one of our hypotheses is that by degrading the homogalactronan, these pectinases can soften the cell wall and allow for the cell to expand more efficiently, or potentially degrade the middle lamella, like we were just talking about, and that results in cell separation. So whenever I walk through Penn State's campus in the fall, I was, see the leaves falling off the trees, and I'm like, so many polygalactronases doing their thing, which probably nobody else is thinking about, but that's okay. So by cleaving pectin, we think that the cellulose and the hemicellulose network in the wall becomes more deformable and able to undergo expansion. That's, again, ultimately driven in part by turgor pressure and also mediated in part by these expansion proteins that disrupt, we think, the interactions between at least cellulose and potentially also hemicellulose molecules. So one of the genes that we've studied in Arabidopsis thaliana, which is kind of our classic model plant, it's like the mouth or the fruit fly of the plant kingdom, is called pectate lyase-like 12 or PLL12. And this affects plant growth. This work was spearheaded by Yintang Chen, who's a former PhD student in my lab, and is now actually a postdoc at Penn. We isolated mutants of PLL12 in which this gene was knocked out. We looked at complementation lines in which we put the PLL12 gene back into the mutant background. We overexpressed PLL12 by a strong promoter. And then we also made some guard cell specific knockdown lines. So we used artificial microRNAs to knock down the expression of PLL12, particularly in the guard cells of these plants. So one of the interesting phenotypes that we saw when we looked at the whole plants is that the PLL12 knockout is very tiny. The overexpression is a bit smaller than our wild type control, which is Columbia over here. When we complement, the plants recover in size to pretty much normal. These are some negative control GFP artificial microRNA knockdown lines that look pretty normal. And then in a couple of our PLL12 knockdown, guard cell specific knockdown lines, the plants are a little bit smaller than wild type. So we then studied the stomatal responses of this PLL12 set of mutants, especially the knockout and overexpression lines, to two treatments that cause either stomatal closure or stomatal opening. So one of the sort of classic drought hormones in plants is called ABA, or abscisic acid, and this causes the stomatal pores to close. So in wild type, which is shown in black here, when we apply ABA to small leaves that contain the guard cells, we see a reduction in the ratio of the pore area to the complex area. We quantified this as a ratio rather than an absolute size of the pore because the PLL12 knockout plants had smaller overall stomatal complexes than wild type. So we see this reduction in the closure of the pore, and you can see that outlined between 0 and 2.5 hours of ABA treatment here. The knockout lines actually start out pretty closed. They open a little bit in response to ABA, which is weird, and then they go back to being pretty much closed. Whereas the overexpression line, the stomatal pores tend to be a little bit larger as a proportion of the total complex, and they don't close as efficiently as wild type. We can conversely use a drug called fusicoxin, or FC, that causes the stomata to open. It actually activates a proton ATPase in the plasma membrane and hyperpolarizes that membrane, causes potassium to enter and makes the guard cells swell and elongate and drives stomatal pore opening. So you can see that the wild type and the overexpression line both have pretty efficient stomatal opening in response to fusicoxin, whereas the knockout, basically the pores don't respond to fusicoxin at all. And then when we look at guard cell specific knockdowns of PLL12, their closure behavior is relatively normal, but the opening behavior shown in blue here is defective as compared to our GFP negative control knockdown lines. So one of the interesting things we did with these mutant and overexpression lines is we studied them using nanoindentation. And this was a collaboration with Joe Turner's lab at University of Nebraska. We exposed the plants to the dark overnight to cause their stomata to close, because plants close their stomata in response to darkness because there's no light to drive photosynthesis. And then we placed them in the nanoindenter machine. And we start with them in the dark for 20 minutes. We turn on the light for an hour, and then we turn the light back off. And by pressing gently on the surface of the stomatal guard cells with this nanoindenter to varying depths, we can get estimates of both the so-called modulus, or roughly the stiffness of the wall, and also the turgor pressure that's inside these cells. So we first press gently at small deformation, so a few hundred nanometers. And that gives us a sense of the wall modulus. And then we press more deeply into the cells, and that gives us a combination of the wall modulus and the turgor pressure. So we're measuring those stiffnesses as the output of the unloading curve of these repeated indentations and bringing the indenture tip back up. And then we put this into a modeling framework where we actually generate a computational model of the geometry of these pairs of guard cells. And we solve that model for both turgor and modulus that gives us the same behavior that we observe when we're measuring these on the nanoindenture, which also has a microscope attached to it. So we can actually watch these cells open and close in response to light and dark. So when we do this in wild type, which is shown in black here, when we start out with the lights off and we turn the lights on, we see a slight increase in wall modulus that slowly relaxes over time and then goes back down to baseline when we turn the lights back off. You can see that the PLL12 cell walls, or the wall modulus in the so-called E1 direction, which is this longitudinal direction, is substantially higher than the modulus in the wild type. And then the overexpression, which is shown in green here, is somewhat in between. When we estimate the turgor pressure based on this approach, we see that turgor pressure jumps up when we turn the lights on in wild type, and then continues to rise until we turn the lights back off when it goes back down toward baseline. And the turgor pressure in the PLL12 knockout is substantially lower than that we observe in the wild type, suggesting that this might be part of the reason that we don't see significant stomatophore opening in response to the treatment that I mentioned earlier, the fusicoxib treatment. So this is giving us a mechanical insight into how the cell and the wall itself are responding mechanically to these light and dark stimuli. And then one of the other exciting things about this set of experiments, which was really one of the first sets of experiments in which we were able to detect the wall modulus and the turgor pressure simultaneously in a physiologically active guard cell, is that these values are changing continuously during stomatal opening and also change dynamically during closure. So it's not simply a switch that goes from sort of low pressure to high pressure and then back to low. It's sort of this gradient that changes over time, which we think is pretty exciting. And then one of the things that we also studied was how the wall structure changed in response to this modification of PLL12. And we saw some subtle differences in staining patterns of different types of pectin that we're able to detect with antibodies as well as probes that bind to the negatively charged residues in pectin. And I'm not going to go into the details of this slide just for time. I want to give a chance for questions and maybe for you to have a bathroom break before the next speaker. But from all these data, we generated a working model for how this particular gene is affecting stomatal dynamics and ultimately plant growth. Where PLL12 is altering the composition of the wall, we think that it's leading to potentially more pectin cross-linking and the stiffening of the wall, which is why we detected a higher modulus in the PLL12 knockout. Whereas in wild type, we have PLL12 tuning the size of the pectin to allow for enough flexibility to allow for stomatal opening and closure. In the overexpression, we think that we're actually probably cleaving the pectin down to a slightly smaller molecular mass, and that allows for the stomata, for instance, to remain open, a little bit more open in response to AB treatment. And one of the interesting things is how we connect the stomatal dynamics ultimately to plant growth. So we're still sort of trying to puzzle our way through that to understand how stomatal function ultimately determines the size of the plant, right? Because it both influences water dynamics as well as the ability of the plant to take up CO2 and generate all those wall polymers that it needs to grow the rest of the plant body. So I'll finish with just one sort of highlight slide showing that by engineering stomatal mechanics, we might be able to enhance plant growth or at least water use efficiency. So this is work from Mike Blatt's lab, in which they engineered a light responsive channel, a direct light responsive channel. So the normal sort of signal transduction pathway from light perception to opening these potassium channels involves a lot of signal transduction steps in guard cells. They sort of short-circuited that by making a channel that itself was responsive to blue light. That's called the blink-1 channel. And when they engineered this into plants and grew them under water limiting conditions, they found that the plants were more efficient at taking up CO2 if they contained this blink-1 channel, which is a pretty cool way to sort of make the stomata respond faster to changes in light availability or water availability, for instance. So stomata, as you saw in the graphs that I was showing you, are relatively slow in their response to things like APA and fusicoxin. They take 2.5 hours to fully respond, which if you imagine a cloud coming over the sun, right, the stomata might be adaptive if they were able to close pretty fast and then open back up once the sun comes back. But they take sort of minutes to hours to respond rather than seconds. So making faster stomata is one of our kind of technological goals in understanding this. Okay, so I'm going to finish up there. That was a very brief and not fully complete introduction to plant cell walls and mechanobiology, but I wanted to give a chance to the audience to ask questions if they have them. So fire away. Or if you all like have to run and use the bathroom, that's okay too.
Charlie, one thing that I actually might be interesting, I gave the lecture on the ECM, the animal ECM yesterday. And do you want to just list, you know, like take 1 minute just to list what you think are some of the major similarities and differences? Because that's one thing we're sort of trying to highlight.
Yeah. So I think major similarities, you know, the ECM and the cell wall are produced by cells, right? So in the case of plant cells, they're produced by the cell that you know is encased in that cell wall, whereas animal cells can produce ECM and then migrate relative to their ECM, although not all of them do that. You know, sort of epidermal cells in many cases in animals produce an ECM and then stay attached to that ECM. I would say one of the major differences is that the animal ECM contains lots of glycoproteins. Those are probably the dominant, what I would call the dominant component of the ECM. Whereas plant cell walls are largely polysaccharide based rather than glycoprotein based. One of the interesting parallels is that certain components of the plant and animal ECMs are calcium cross-linkable. So we have calcium cross-links that happen between cadherin molecules, for instance, in certain animal ECMs. And pectins are cross-linkable by calcium as well. One of the other interesting parallels is that both animals and plants have mechanisms for monitoring the mechanical status of the ECM. So you can think about, you know, for instance, the integrin system in animal cells being able to monitor and respond to the mechanical status of the ECM. Plant cells, we think, have mechanosensation in the sense that they can, they have receptor-like kinases and that are potentially linked to channels and stretch channels that allow them to respond to the mechanical status of the wall. Although that's different, it's a different mechanism because they don't have integrins in the same way that in focal adhesions, the same way that animal cells do. So there are a lot of parallels in terms of sort of the functionality of the plant and animal ECM. It's just that the components are slightly different because they're, you know, diverged over the last whatever billion years or so.
Sort of an interesting, you know, the idea of convergent evolution. It just tells you something interesting about how important these functions are.
Yep, it's like bats and birds, right?
Yeah.